Good morning, everyone. My name is Elisa Corbin, and I'm an assistant professor at the University of Delaware in the Biomedical Engineering Department. Um, so today I'm going to talk to you about some dynamic microenvironments that my lab builds, as well as some of the questions that we're targeting to answer. Specifically, we're interested in cardiomyocyte mechanosensitivity. So the heart is a dynamic pump subjected to essential mechanical load during each and every heartbeat. And this is really the core concept of our research. More specifically, my lab's overall research direction is in advancing engineering technologies to study how these dynamic cellular processes, with a specific focus on mechanics, modulate both biochemical as well as pharmacological responses. So what my talk really boils down to is how form and function interact dynamically, and how we go about mimicking the cell's natural environment. So currently, there exists two really well-accepted models in biology, right? Animal models and in vitro models. Animal models can teach us a lot about what happens in the complex organism. However, we can't necessarily exert the same kind of control that we can in vitro. Whereas in vitro, we have this full control over all these parameters, but we still struggle to recapitulate what happens in vivo. So me being an engineer, I look at the problem and I say, we need to be able to create better in vitro models. So what parameters do we actually need to consider? Well, we know that cells can feel the mechanical environment, mechanical properties of their environment, and we know that these mechanical signals can drastically influence how a cell and tissue behave. So what we need to do is create tools with the capabilities to control these different mechanical properties. So I work with these Magnetorheological elastomers, which provide tunable and reversible stiffness control. Maybe you've seen them in different large-scale mechanical systems. They're used in actuators and pumps. They're usually really hard and stiff. And for ours, what we did was we actually combined a really soft polymer, PDMS, and combined iron particles randomly oriented within that matrix. And then by simply applying a magnetic field, we can achieve changes in elastic modulus. In our case, we use simple permanent magnets, and then adjust the distance of that magnet to adjust that field strength. And we can then modulate that elastic modulus. And we have control of this modulus between about 10 kilopascals to 50 kilopascals, which is really ideal for cardiovascular physiology, where 10 kilopascals being for sort of a normal adult myocardium, and 50 kilopascals being closer to the pathologic adult myocardium. So with these materials with bidirectional control, we can start to add a temporal dimension to how we investigate the response of these cells to substrate stiffness. Right? We can ramp up the stiffness, we can ramp down the stiffness, we can cycle that stiffness at different frequencies, all in the same dish. So what I'm showing here on this slide are really two canonical markers of mechanosensitive processes. What better way to do this than through sort of cell spreading, and where we know that cells spread more on higher stiffnesses. And what we're showing here is that there's this acute reversibility. This is over, I think, 18 hours that we see this spreading and then shrinking again of these cells. We also looked at YAP translocation. YAP is the yes-associated protein, and this is, again, sensitive to stiffness. And what we see is that the stiffer the substrate, the more YAP translocates into that nucleus. And again, we see this sort of acute reversibility. But, let me come back to our goal is to really create a better in vitro model. And 2D models will only get us so far. So I want to start by showing you guys some of the tissues that we build in my lab. These consist of 95% induced pluripotent stem cell cardiomyocytes, and 5% cardiac fibroblasts. And as you can see here, the cells actually beat spontaneously. But we can also electrically pace them with a field stimuli. So maybe you're asking, what does this device actually look like? Here are a couple of SEM images showing what these devices look like. It consists of really two tiny pillars with really small caps on top inside a larger well. And what we do is we take our cells and mix that into a collagen matrix, spin those cells down into the well, add fresh media, and these tissues will actually self-assemble over about 24 hours. Right? And as they self-assemble, it gains tension against those posts, causing them to bend. And we can use then simple high school physics here in order to determine that force generation. So I like to show this video. Most people don't quite visualize what that self-assembly process looks like. But what you can see here is that there's a correlation of tension sort of generated over time as this sort of system sort of comes together. And it's pretty interesting. It's really an energy conservation problem. But for engineered cardiac tissue systems like this, being able to dynamically model responses to increases in, say, afterload, where afterload is the force that must be overcome before a muscle can actually shorten, this increase in afterload is clinically relevant in pathological cardiac remodeling. So what I did was we actually intersected these magnetorheological elastomers that we were showing in 2D with this microtissue model to ultimately make these vertical posts that magnetically stiffen. So what I'm showing here is the change in force generation detected both from a pharmacological side as well as just mechanical influencers, right? And I hope this shows you how important sort of the mechanical side of things are really can really drive different changes in biology. Which this brings me to my last slide, which is on my INBRE project, where we're trying to model one of the common treatment options for sepsis. And sepsis is a life-threatening organ dysfunction that is caused by a dysregulated response to an infection, any infection. And sepsis can manifest itself in many ways, making it extremely challenging to diagnose and to treat. Additionally, because time is really of the essence in treatment. So my project is to take these cardiac tissues and treat them with septic serum to induce a change in the contraction kinetics. And then hopefully by applying mechanical property changes as a therapy, we can try to mimic, say, an increase in afterload that has a clinical proxy of vasopressors. So ultimately what we're looking for are correlations between contraction responses as well as different biomolecules. So couldn't do this alone. I want to just point out a couple people from my group, my collaborators at Bryn Mawr, as well as University of Pennsylvania. And this work has been sponsored both by Excel and INBRE. With that, I can take any questions.